So what we're doing now, we're just starting a clinical trial using what are called gene-edited iPSC-derived MSCs embedded in 3D bioprinted hydrogel scaffolds. And so essentially what that means is that these iPSC cells are genetically engineered because there's a Nobel Prize from Japan. He's called Professor Yamanaka. He won the Nobel Prize for what are called the Yamanaka factors. He basically figured out that you could take any cell. The Japanese are so smart. So and that's why I worked in Japan this year and learned a ton. But they figured out that you can take any cell in your body and you can overexpress these 4 transcription factors and you can turn them back into a baby stem cell. How crazy is that? That your body has a memory to, that's what I mean. Your body has this memory to heal itself. And we're just trying to unravel that mystery.
One of the big questions I wanted to kind of dig into, or big topics I want to dig into, and it seems like it's pertinent to kind of break way into the topic now, is ethics, right? The big thing with stem cell, what was Christopher Reeves, was it the original Superman? He ended up with like a spinal cord issue, and there was like a big advocate. He was an advocate for stem cell research, if I'm getting that story right. But then there was like, you kind of ran into like the abortion crowd at the same time, and you're like, this is weird. Why is a guy in a wheelchair pitting against mothers from the church group? Like, this is nuts. And there was always the conversation of like, okay, the ethical harvesting of stem cells, and you use the word manufacturing a lot, which is like a really interesting word to use for what was previously seemingly like an extraction process, right? And so I want to talk about the ethics specifically around stem cells first. And then you've used the word editing and engineering a handful of times. And the last guy to use a word like that was God, to whatever one you believe. Like, I'd be really curious as to like how innovation runs alongside ethics when you guys are playing God.
There is a lot of ethical issues to consider. So the first one to address is obviously, are these cells being sourced ethically, meaning are any embryos being harmed? So no, we're not sourcing them from embryonic tissue. The reason during Reeves' time, there was only embryonic stem cells at that point, because embryonic stem cells are from the fetus, right? They're from the embryos. And they're obviously the most potent because they're what's called totipotent, which means they have a ability to turn into any type of tissue in your body. And they also have an ability to regrow new tissue. They have more stemness than mesenchymal stem cells, which are derived from umbilical cord tissue. So meaning they have a more ability to proliferate and grow new tissue. But then the risk of that is that tumors and cancer.
Bone.
Exactly, eye bone.
Right.
Exactly. And we talked about that last time. Why did that happen? Because that was embryonic stem cells. And so they have too much stemness, and that can lead to unforeseen circumstances, like unforeseen consequences. And back then, because they only had embryonic, they were kind of like, well, you're going to be hurting embryos. Like, you can't do that. So people transitioned over into umbilical cord tissue, mesenchymal, because mesenchymal, they're not as strong as embryonic, but they still have good properties in terms of reducing inflammation. They can treat like soft tissue injuries, autoimmune conditions, but they're not really regrowing new tissue. So now with the genetic era, let's call it genomic era, we can actually edit those Yamanaka factors into a cell, and it's called an induced pluripotent stem cell, iPSC cell. And the iPSC cell has the same stemness and the same properties as an embryonic stem cell. And you can just take it from a skin tissue biopsy.
So you're just regressing it down to its
Exactly.
starting point.
Exactly.
I mean, that to me, like a very surface level, like that's not just longevity. Like that sounds like a reversal process, because like the process of synthesizing that, I don't know if that's the right term, back to embryonic, it seems to defy. It defies some sort of medical principle or some sort of.
No one thought that was possible. When Yamanaka did it, everyone was just like, that's insane. That's why he won the Nobel Prize.
Right, I guess
it was
so.
Just like, this is unbelievable. And it's still hard to appreciate how big of a discovery that is, because it's basically like saying your body, wait a minute, I can take your old ass, like you can be 60 years old, I can take your old cell and I can make it into a baby stem cell. Like, that's wild.
Yeah.
Because that then opens up the box for saying, wait a minute, maybe we can cure aging, or maybe we can reverse aging, or maybe we can just stop it altogether. So that's where this is all headed. And that's why, but then to the point about iPSC cells, as we were talking about embryonic cells, because they're so strong, they can also have uncontrolled proliferation. So the technology we have we basically gene edit these iPSC cells so it has a kill switch to prevent uncontrolled proliferation. And then they're also gene edited with the cloaking mechanism. So that's what we're using now.